Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics. Most people think that vitamin D is something that supports bone health or maybe helps the immune system, but its role in cancer biology may go much deeper than that. What if vitamin D doesn't just support cancer treatment, but it actually dismantles the way that cancer cells use glucose and glutamine? In this video, we're going to look at how vitamin D can influence cancer at a metabolic level, specifically how it affects the cancer cell's use of glucose and glutamine, the two primary fuel sources. These pathways are central to cancer cell growth and survival, and disrupting them can fundamentally change how these cells behave. So without further ado, let's get into it. So this video is going to be about not if, but how does vitamin D kill cancer cells? And we're specifically going to look at molecular mechanisms. And before I get into vitamin D specifically, I'm going to look at this graphic from the C-Free group and get us oriented about why I picked those specific papers in this video to present vitamin D in that way, because I want to bring us back to the fundamentals of metabolic therapy. And metabolic therapy is about leveraging metabolic differences that cancer cells have or don't have compared to normal cells and what they have or don't have. And one of the things that I hope we've established at this point is that cancer cells will hyperutilize both glucose and the amino acid glutamine on the order of about 10 to 30-fold more than their normal cell counterparts. And so part of our job in metabolic therapies is to find strategies to either limit glucose or glutamine availability, to lower glucose from getting into a cell through some kind of a transporter, or glutamine into the cell through a transporter, and then the metabolism of glucose through glycolysis and subsequently lactic acid fermentation, and glutamine through glutaminolysis and the fermentation of glutamine into succinate. And that's why lactic acid here and succinic acid here are at the bottom. And so when I look at a compound in terms of a proof of concept, does this make sense that it would be effective potentially against this disease? Then I'm going to look at it through the lens of the way that it sustains itself and protects itself. And so that's how I'm looking at vitamin D specifically in this video. So I'm going to start out with this paper that was published in June of 2022, and it was titled Vitamin D Regulation of Energy Metabolism in Cancer. And it says here, I highlighted this portion of the abstract where it says, however, recent evidence has identified 1,25-hydroxy-vitamin D, or calcitriol, or activated vitamin D, as a regulator of energy metabolism in cancer cells, where requirements for specific energy sources at different stages of progression are dramatically altered. The literature suggests that 1,25-hydroxy-vitamin D regulates energy metabolism, including glucose, glutamine, and lipid metabolism during cancer progression, as well as oxidative stress protection, as well as is closely associated with energy metabolism. And so this paper had a graphic associated with it where we see 1,25-hydroxy vitamin D binding to the vitamin D receptor, and that being basically transitioned to the nucleus, where it's then having, effects on genes at the level of the genes and on target kind of metabolism changes. And what we're saying here is that this will ultimately lead to less glucose metabolism, less glutamine metabolism, less lipid metabolism. I'm going to unpack what I mean by that. It does not mean that these cancer cells are using fat for fuel. And it decreases the oxidative stress protection for these cancer cells specifically, which then leads to decrease in cancer progression and survival of cancer, not of people. So let's unpack why this is interesting. Well, vitamin D by itself is not necessarily an antioxidant, but it functions through at least two major antioxidant response elements, one of which is called the NRF2 pathway, and one of which is called the Klotho pathway. And those two antioxidant response elements are generally going to have a pro-antioxidant effect. They will upregulate endogenous antioxidant systems. So when vitamin D is having a decreased oxidative stress protection in these cancer cells, that is particular because in normal cells, it would have a opposite effect. Within cancer cells, it actually strips the cancer cell of protection from oxidative stress. So let's look specifically at some of these key targets. So here is glucose at the outside of the cell and has to be transported in through a GLUT transporter. And then that glucose has to be metabolized through a process called glycolysis. This is the breakdown of sugar. And the end product of that is generally going to be lactate. So we see there that D3 has diverse effects on multiple systems. Everywhere you see a little line with a little stopper on it, it's showing that it's having an effect at those levels. And then you're also going to see that it has effects at the level of glutamine. And where it has effects at the level of glutamine is both in glutamine transporters, such as SLC1A5, or in the glutaminase enzyme GLS. Now, I think this is important to note. What is happening here at these sites is likely not a direct blockade. What's happening here is that when vitamin D is active in the cell, it's actually downregulating the overexpression of these proteins, right? So this protein comes from an RNA and then, before that a DNA. And so what vitamin D is doing is it's not necessarily blocking this transporter directly. It's that it's cutting off the cancer cell's ability to make this protein to begin with. The same with GLS or some of these other enzymes. And this is the idea of fatty acids, of what it's doing here. So we're not really concerned about fatty acids being brought into the mitochondria for energy production. You have to have an intact mitochondria and you have to have oxygen for that to happen. But that doesn't mean fatty acids are not used to make other fatty acids, or even glucose and glutamine to some degree are being used to make citrate, and then citrate is being used to make fatty acids. And those fatty acids then contribute to the raw materials of the cell. And vitamin D has effects at the level of the fatty acid synthesis as well, or metabolism is kind of a more broad way of talking about fatty acids. Now, let's take a look at exactly where in these processes vitamin D has effects. So we're going to start with the glucose system glycolysis. So we see here everywhere I've highlighted a red, that's where vitamin D is at, right? So here's a GLUT transporter, right? Here's hexokinase, here's PFKFB, here's lactate dehydrogenase, here's these lactate and ketone transporters, the MCT, monocarboxylate transporters, and then PDK inhibitors as well. And so if I was to put that on a graphic, so the graphic is this, I've added arrows and where vitamin D is acting. You see here that vitamin D is working on glucose transporters at the level of glycolysis at multiple levels, right? PDK, similar to that of dichloroacetate, hypoxia-inducible factors, and lactate transporters. So as I've said before, vitamin D is a rock star at being able to collapse the Warburg metabolism or the substrate level phosphorylation of glucose, which is what Seifried et al. Are using instead of Warburg effect. Now let's take a look at glutamine. You see here that 1,25-hydroxyvitamin D, this is what's mostly studied, so this is where the majority of the literature is at, has effects at the level of glutaminase, glutamine synthetase, glutamine dehydrogenase, glutamine uptake ports, as well as another important piece of this story, which is SLC711, which is closely related to glucose and cysteine transport and glutathione production for cancer cells specifically. It makes a heck of a lot more sense if we look at it on a graphic like this, where I put arrows. So here's the glutamine transporter that's affected by vitamin D. Here's the glutamine enzyme that's affected by vitamin D. This, to orient us, is the same enzyme that is blocked directly by DON. Here is glutamine dehydrogenase affected by vitamin D. This is what is directly blocked by EGCG. And this is vitamin D. And this is SLC711, which is the transporter that is related to glutathione synthesis and redox homeostasis, where glutamate is exported and cysteine is brought in. This is what is blocked directly by sulfasalazine, aristin, etc. Vitamin D is decreasing the expression of this protein. So you can see here that not only is vitamin D, and this is just the active vitamin D, there could be other off-target effects from these vitamin D, D3 and L3 hydroxy derivatives, which are understudied. But this is the activated calcitriol molecule, vitamin D, 1,25-hydroxyvitamin D. So we see how it's collapsing glucose metabolism. We see how it's collapsing glutamine metabolism, as well as the ability for the cancer cell to make glutathione through this SLC711, also known as XC. It does other things also. There's a whole host of cancer-related signaling pathways that are affected by 1,25-hydroxyvitamin D, as well as the D3 and L3 hydroxy derivatives. And then there are a whole host of anti-inflammatory and other effects. I don't even have enough time in this video to go over all the things that vitamin D can do to help us fight cancer, some of which is inflammatory related, as well as off-target non-glucose and glutamine related targets that also have clinical benefit. So I hope you can see that vitamin D is a cancer fighting powerhouse. It affects and dismantles the glucose uptake and utilization system, and it dismantles the glutamine uptake and utilization system, as well as the ability for the cancer cell to make glutathione. So this is a very important video to highlight not if, but how vitamin D destroys cancer cells. And if you like videos like this, please like, share, and subscribe. And until next time.